Okay, so hello everyone. Let's continue where we left off last time. Just a little bit of a recap. Like always, can someone state briefly how do we measure total carbohydrates? Yes, common. Huh? Yes, so when we're looking at total carbohydrate in a food product, we just do all approximate analysis and we subtract from 100 and we get total carbohydrate. But if I have a simple beverage matrix or a purified solution with only total carbohydrate, we do the phenol sulfuric acid method, which we would do in a lab normally, but we'll have a virtual lab in a couple of weeks and talk about how we do it in the lab. So it's based on breaking down the carbohydrates into monomers, and in the presence of acid and heat, they convert to furans, and in the presence of a phenol, the furan will react with the phenol and give you a chromophore that we would measure spectrophotometry. Okay, so what are the preparatory steps for carbohydrate analysis? Yes, Dylan. Yes, it is absolutely drying and fat extraction. First, it's good to say and safe to say homogenization of your sample, selecting a representative sample, then dehydration or removal of moisture, and defatting, removal of fat either by batch extraction with a solvent or success extraction. Then you have a dehydrated, defatted sample ready to do carbohydrate analysis. If we're looking at mono and di and oligosaccharides, then we need to separate them from starches and protein. We solubilize with the hot 80% ethanol solution, alcohol solution. Then your sugars, small sugars will solubilize, and starch, protein, and fiber will precipitate. So that actually answers the third question. So we haven't gone through all of the methods yet for monodi and oligosaccharides, but we talked about the reducing sugar methods, which are based on reducing of cupric ion to cuprous. So we have several. We talked about Smuggey-Nelson as one example, but they are based on the ability of the reducing sugars to reduce cupric to cuprous, and the cuprous reduces arsenomilobdate reagent in the Smuggey-Nelson and gives you a specific color that we measure spectrophotometrically again. All right, so that's where we were, and I stopped at the enzyme. Let's see here. Here I'm just showing the structure of a protein. So here, enzymes are proteins, basically, and they are biological catalysts. They can be used as biological catalysts. And the way we use them, actually, that your constituents of interest are the substrates. So if we want to analyze for glucose, we need to use an enzyme specific for glucose. If we want to analyze for a disaccharide, maltose, for example, or fructose, again, we need to use a specific enzyme that would hydrolyze that specific mono or diglyceride. There are usually very specific enzymes for the substrate of interest. So you will have your reactant being your substrate, you have the enzyme, and then you have a product. And the product often is a chromophore. And we measure accordingly the absorption of light of that chromophore. Sometimes we need to have, okay, I'm not sure why I'm not getting my slides to move. Okay. Sometimes we have a coupled reaction, we call it. So two enzymatic reactions. The first one is the auxiliary reaction or the measuring reaction, where we have our food constituent and a specific enzyme and a co-reactor, and it gives you a product. That product is still not a chromophore. We need a chromophore. Then we take that product and react it with another components of the substrate and have another enzyme, and we get a reaction called indicator reaction. It's called indicator reaction because the product from this reaction is potentially the chromophore. So here what this is showing is the R or S, which are products in this case, could be the chromophore that we measure, or the compound here that reacts with the first product we get, that's P, the first product, this could be a chromophore. And when it reacts with this product, it might lose its chromophore ability. So we see a reduction in absorption. So sometimes you measure a change in absorption that is increase in absorption. Sometimes you measure a change in absorption that is decrease in absorption. So if C is the chromophore, then we're going to see a decrease in absorption. If R or S are the chromophores, then we can see an increase in absorption. So an example of this, let's see here. Yeah, this is just showing you that auxiliary reaction can also be called measuring reaction. The second reaction is called indicator reaction because it gives you a chromophore. That's where you measure absorption. So this is an example of a coupled reaction for measuring glucose specifically. In this case, we have glucose reacting with ATP, and in the presence of a hexokinase enzyme. The hexokinase is going to take a phosphate group from ATP and give it to the glucose. You end up with a glucose 6-phosphate, so where the phosphate is at the carbon number 6, so glucose 6-phosphate and ADP, because we lost one phosphate. So that is your first reaction, okay, auxiliary reaction or measuring reaction. Glucose 6-phosphate, we reacted with NADP in the presence of glucose 6-phosphate dehydrogenase. So this enzyme is going to take out the hydrogen from glucose 6-phosphate and give it to the NADP. Then you get NADPH, and this one absorbs light. So we can measure the absorption of NADPH, which absorbs light in this region, 340 or something around that. So the application in mono and di and oligosaccharides are many. So we can measure monosaccharides, fructose, glucose, galactose, or oligosaccharides, disaccharides, specifically lactose, maltose, and sucrose. There are specific enzyme assays for those. And we can use enzymatic methods to actually measure starch, total starch, and specifically amylose and amylopectin. We can get the ratio of amylose and amylopectin using enzymatic methods. And we'll talk about using enzymes for measuring starch later. So a lot of advantages. They're specific usually for the substance we are measuring. So they're specific for glucose, if we're measuring glucose, or specific for fructose. Kits are commercially available. They are very easy. So we get a kit and it has the enzyme, it has the necessary substrate, and it has our standard. So if we're measuring glucose, we get a glucose standard and we get the reagents and even the steps. So it just tells us what to do and we just mix in, complete the reaction. And it generally have low detection limits. That means it has low thresholds, so we can detect concentrations at even low and diluted concentrations. Some of the disadvantages, depending on the wavelength that we're measuring in, there might be components in our sample matrix that absorb in the same range. So that is a potential problem. Another, I said here usually specific, but not always 100% specific. Why is that? Because it depends on the enzyme purity. Sometimes the enzyme is not very pure, the one we purchase, and sometimes it is pure. So that's why we say it's not 100% specific. And sometimes if you have monomers that are very close in structure, the enzyme might also react with that. So that is another potential issue. So this concludes it for enzymatic methods. We have a whole chapter on enzymes or utilization of enzymes in food analysis. We'll talk about enzymes in more details there in a couple of weeks. So other methods are chromatographic methods. So we'll spend some time going over chromatographic methods of analysis. So at the beginning of the year, we talked about chromatography, and I told you I'm covering basics, but then I will start giving you examples later on when we start covering specific chapters. So here comes chromatography again. And in this case, I'm going to give you chromatographic methods examples specific for monodi and oligosaccharides. So if we're looking at qualitative methods, just screening qualitative methods to look at different components in our sample, we can do paper chromatography or thin layer chromatography. Sometimes thin layer chromatography can be used for quantitation or semi-quantitation. If we run standards along the samples, we can measure the intensity of the spot after we run and develop our thin layer chromatography plate. Or we can scrap off the spot and clean it off, remove the resin, and do a spectrophotometric method for quantification. That means after we purify, let's say, the glucose, we can quantitate it by taking that spot off the plate and doing an enzymatic assay. Thin layer chromatography, we can use cellulose. Most common, it is silica. The specific organic mobile phase is listed here as FYI. Compared to paper chromatography, if you remember, you get better separation, better resolution, and the spots are sharper. Okay, but if we want to do quantitation and not just screening, oftentimes we do high performance liquid chromatography. This is much more common for mono and di and oligosaccharides analysis than paper and thin layer chromatography. So here we have 4 different ways of utilizing HPLC to determine mono, di, and oligosaccharides. So normal phase, reverse phase, anion exchange, and cation exchange. So before I dive into all of these or individual ones, let's just try to remember our basics here, because I want you to know the basics before going into the details here. So let's try to remember what is normal phase HPLC. Okay, I have just few students here and the rest online. Coleman, do you want to? Yes, so Coleman told us and saying here a couple of folks, polar stationary phase and non-polar mobile phase. So your constituents will be binding via relative polarity to your normal phase and separating that way. So the least polar will come out first and the most polar will come out last. Very good. And reverse phase is the opposite, so that I don't have you spend time on that. So it is really the opposite. So And it's called reverse phase because normal phase was used first, and then they utilized reverse phase, so they called it reverse phase. So they inverted everything. So the stationary phase is your nonpolar, your mobile phase is polar, and your molecules are separated also based on relative polarity, but in this case, most polar will come out first. Good. So what is anion exchange HPLC then? Yes, so anion exchange binds anions to the stationary phase. Exactly. So when you see anion, you know that your components are negatively charged. So you look at the first word and you go anion exchange. So I'm exchanging negatively charged components. So my stationary phase is positively charged so that it can interact and exchange negatively charged components. Cation exchange is the opposite. So I am now exchanging um, positively charged compounds, and my column is negatively charged, or the functional groups on my column are negatively charged. Okay, so looking at this, carbohydrates, mono, di, and oligosaccharides, are they charged molecules? Can they be charged? Common, you say no. They can. Normally, if we look at pH 7, carbohydrates, they're not. So glucose, fructose, sucrose, they are not charged molecules. So how are we going to use anion exchange and cation exchange to separate mono and diglycerides or oligosaccharides even that are not charged? There are ways, and we're going to talk about that. Okay, so we'll start with normal phase. So your stationary phase is polar. So we first of all we think, okay, unmodified silica. Silica can be polar, and then your carbohydrates, the monodienoligosaccharides, will interact very strongly with this selenol groups, silica functional groups. So if they interact very strongly, they will have very similar interactions. They will not separate. We will have the hardest time separating them. It's almost impossible because they are going to interact very strongly. And similar monosaccharides, similar in structure, and disaccharides are going to, you'll not be able to separate glucose from fructose or uh, maltose from sucrose. Okay, so what you would do is, in this case, you chemically modify the silica. So this would be called HILIC, hydrophilic interaction liquid chromatography. Earlier, when we were talking about chromatography, we covered HIC, which is hydrophobic interaction chromatography. Here, normal phase using modified silica is called hydrophilic interaction liquid chromatography. So what is the modified silica in this case? In this particular case, there are many different helics, but in this particular case, the silica groups are modified with amine group. So you see here your silica functional group. The end of it, you have, well, you would have a molecule that has an amine group that we get it attached to the silica. So now you have an H2 at the end here. So your carbohydrates, your mono, di, and oligosaccharides have OH groups on them. So the OH group will react with an H2 groups by hydrogen bonding. So in this case, in this particular case, hydrogen bonding is the type of interaction that happens. So, all right, if it is interaction with OH groups, I'm going to ask you a question before you look to the next slide. Just stay focused with me. Don't look on the next slide and tell me, If you have glucose versus sucrose, which one will leave the column first? Based on knowing that the OH groups interact with NH2 via hydrogen bonding. Which will come out first? Glucose, common? Glucose will come out first. Why common? And yes, so it's a smaller molecule and it has less number of OH groups. So in this case, yes, I see many of you answered glucose. In this case, glucose will interact the least and will leave the column first. Actually, if you have a fructose and glucose, see what happens. Fructose come out first because fructose is a ketone group and glucose is a hexose. So fructose will have less OH groups to interact with the NH2, so it comes out first. And glucose next, and a saccharose, lactose, and so on. So here the interaction is the hydrogen bond depending on how many hydroxyl groups are available for interaction. All right, so in that case, we have the amine bonded stationary phase, and we have detection usually with refractive index detection. In this case, because we're using refractive index detection, we cannot use gradient dilution because if you remember that detector based on refractive index, and if your gradient is changing over time, your refractive index will change. So it won't be constant. So that makes it difficult for quantitating the actual components leaving the column because it is simply based on the refractive index of the solids or the components in your mobile phase. So it is not the greatest detector in terms of sensitivity, and it's not very specific. So if you have good separation and high concentration, it's a good detector. So there is a limitation with the helic for the amine bonded stationary phase because sometimes or many times you have your reducing sugar. So monosaccharides are reducing sugar. In this case, what can happen is a Maillard reaction. So over one potential reaction is the glucose reducing, reacting with the amine group on the column and forming a shift base. And that potentially kind of blocks your amine group from reacting with the sugars. So it happens over time. The more you're injecting, the more samples going through, the more amine groups will be either cleaved off or blocked. To compensate for that, you add amine groups in your mobile phase. So you make sure you continuously add what do you call them? Modifiers. So you add modifiers that have amine groups. So you continuously regenerate your column. So when you hear the word regeneration, it applies not just for this, it applies whenever you are regenerating functional groups in your column. There are other modifications for analysis of carbohydrate, not just utilizing of amine groups. There's diol bonded, cyano bonded, stationary phases. There are all different types of columns that you can find for normal phase HPLC of monodioligosaccarides. Reverse phase. Now here we have a different problem. In reverse phase, all of your polar compounds will come out first. So here you have a really hard time separating monosaccharides. So they will all come out potentially with almost with your solvent front. They're very polar, they're not going to interact with the column. So what you do in this case, your mobile phase is water. So to solve this problem, if we only have reverse phase and we want to run reverse phase, we can add salt to your mobile phase. By adding salt to the mobile phase and to the actual preparation of your sample, then you can enhance interaction with a nonpolar stationary phase. The other problem that you face with reverse phase chromatography is the formation of anomers. Remember from your chemistry, general chemistry, anomers is just basically, if you look here, where's the OH group? Here's the OH group here, whereas the OH group is down here. So here you have OH right here and H here. This one you have the OH up there and the H here. So it's just geometric different location of your OH. So sometimes you get that happening and you see a split in the peak. To prevent splitting in the peak, we add amine groups to the mobile phase that accelerates the anomerization. So all of them will be in one form of anomer, then there won't be a split in the peak. Anion exchange. So here your functional group on the column is positively charged. And in order to make your carbohydrate negatively charged, you run them under really high alkaline conditions. So your OH groups will ionize. Under really high alkaline conditions, the OH group will give up its hydrogen. It will become O minus. And that happens at really high alkalinity, 10 or 11 pH of 10 or 11. So now your OH groups become O minus. So again, what's the order of elution in this case? Monomers first, dimers, or oligosaccharides? Coleman? Monomers first. Again, the same concept. The more OH groups we have, in this case, there will be more ionizable groups. They will interact more with the column. And the less OH groups you have, the less interaction with the column because you will have less O minus. Because it is going to be charged, a common detector is the PAD, which is a pulsed ampliturometric detection, or it is a type of pulsed electrochemical detector. And the order of elution is monomers, dimers, and so on. That's an example of the sugars in molasses and their order of elution. Now, cation exchange. So now we need to make your sugar positive. There's no way you can make your sugars positively charged. So we can make them negatively charged by changing the alkalinity of the solution, but here we can't make them positively charged. The one way to separate using cation exchange chromatography is adding counter ions. So counter ions that are positively charged and can react with the OH groups via coordination complexes. So they will react here with the positively charged counter ion, and that positively charged counter ion will then interact as well with the column. The difference here in cation exchange compared to anion exchange, you also have size exclusion embedded in this chromatography. So they are separated based on size as well. So they're separated based on size in addition to the positive charge from the counter ion. So we see here in this case, sucrose elutes first because it's larger. It's a larger molecule than glucose, and fructose is a ketone, so it assumes a smaller ring structure than glucose. So the mobile phase is water, and also having your counter ions. Diluted acids can be used, like HCl, for example, and the hydrogen or the proton from the acid can be the counter ion in this case. Okay, we can use gas chromatography as well. So in gas chromatography, now we have a different situation. We
Yeah,
need to, yes?
Chrissy had a question about why you would go to the extra effort of using cation exchange.
Oh, it's not, yes. So why do we, I was actually going to talk about that after finishing chromatography, is that in chromatography you have so many different options, and it depends on the components you are looking to separate. Sometimes you have better separation using cation than normal phase, depending on the different individual components. So you decide based on what's published, what's the sample matrix you have, and also what you have available in your lab. So it's many different combination. You have so many different methods, but that sometimes one is better than the other. Sometimes you go simply with what's available. You have a column, a specific column in your lab. You have a method already, an SOP, standard operating procedure. So you would go with that. Um, yes. Yeah, I saw it, Brigida. I saw it. Okay, so there is explain cation dilution order again. Yes, so if I go back once then, so the elution here is based on ion exchange and size exclusion. So if you see here, we have size exclusion and ion exchange. So the column, the stationary phase, has two embedded features. Size exclusion, so you have resin that is polymerized and you have certain pore sizes within the particles that allow gel filtration. Basically, you're filtering based on size. And at the same time, you have the ability to separate based on charge as well. So you have those resin polymers, they have charged groups that are negatively charged. So you would have interaction with the counter ion that form coordination complex with the sugar, and they go through based on their size. So the smaller ones go through the pores more and get delayed, and the larger one have less access to the different pores and come out first. So hence here in this case, you have the sucrose, which is bigger than glucose, and glucose is geometrically bigger than fructose due to being a hexose versus ketose. Does that answer? Okay, thank you. Okay, so moving to gas chromatography. So in gas chromatography, in this case, you need your compound to be thermally stable and volatile. And sugars are not volatile, but alone they're not thermally stable either. So what can we do to analyze carbohydrate by GC? The answer is on the next slide. Derivatization. The same what you did with fatty acid analysis. Fatty acids are not volatile. We turn them into fatty acid methyl esters. In this case, we have the sugars. We can derivatize them by silylation and acetylation, either or. But there are times where you want to use acetylation if you're looking for monosaccharides, and this is done quite a bit when we want to characterize fiber. So later on, when I talk about fiber, if you want to know the components of the fibers that you have, you break down the fibers into the monomers and you do acetylation and you identify the monomers in your fiber. And this way you can determine, okay, I have hemicellulose, I have cellulose, I have pectin, what do I have? Okay, so here's what happens during derivatization. First, you start with reduction. You reduce the sugars into alcohol, and then you react the alcohol with acetic anhydride to form an acetate. And that is what is thermally stable and volatile. Again, here, this is just an example of using GCFID to determine the monomers. Here you have mannose, glucose, galactose, different monomers, and you have internal standards there. Okay, so again, you need to know that you have options. There's so many options, HPLC, GC, and within HPLC, many options. It depends on your sample, the different components you're trying to analyze, the availability of the method, and availability of the instrument and columns. So you have so many different things to consider to choose a method over another. Okay, so that concludes analysis of mono, di, and oligosaccharides. Now let's go to the bigger carbohydrates, the polymers, starch and dietary fiber. So with starch, we either look at total starch content, or we are looking for ratio of amylose to amylopectin. It's important, the ratio is important for functionality. Is this type of starch going to give us a good pasta, or this type of starch is good for viscosity? So it really depends. So the amylose to amylopectin ratio is very, very important, as well as total starch. It's, they all relate to the functionality of our ingredients, for example, that we use in different formulations. Again, degree of gelatinization and degree of retrogradation is important. It's an indication, for example, of digestibility and, let's say, the glycemic index. So if you have high degree of starch gelatinization, you potentially have a very digestible starch and potentially a high degree of or a high glycemic index. Pastas, for example, because you have a high level of retrogradation, they say if you eat pasta, it's probably better than eating rice if you are diabetic. That's because you have more retrograded starch in pasta than in rice. So that's why it's important to know these different characteristics of starch. That's just an example. There's so many other reasons, but these are only examples. So you can measure starch qualitatively. If you want to know, do you have starch in your sample or you don't have starch? Just as simple as that. You can add iodine. Iodine reacts with the starch and gives you a blue color. We sometimes in a reaction that involves iodine, we use starch as an indicator. And we'll have that in the following lab, actually, when we talk about fat characterization and different methods. Sometimes starch is used as an indicator, gives you that blue color for a reaction that involves iodine. But in this case, it just tells you if there's starch or not. It just gives you that blue color. You can look at starch characteristics of the granules. So the granules of the starch. You can do microscopy to look at different granules of the starch. Then that's more looking at physical structure and the impact. Let's say if you want to look at starch damage, the impact of milling and changes in the starch characteristics or the starch granules. And also if you want to look at differences in starch granules between different grain varieties. So there are just, these are qualitative methods. Then you have the quantitative methods, and almost exclusively the quantitative methods for total starch or amylose amylopectin are based on enzymatic assays. Okay, so quantitative analysis, as I said, by enzymatic methods. So the starch is converted totally into glucose because starch monomers are glucose. But starch is complicated. You have branches in the starch, and you have large molecule in the starch. So you need multiple enzymes to break that starch into glucose. So here is a slide that you'll probably seen somewhere else in your chemistry lectures, perhaps. So here you have the starch molecule, and it's just summarizing the different enzymes that can take have role in breaking down the starch into glucose. So I want you to know the difference between these enzymes. You have alpha amylase, which is a very common enzyme, and you'll find it in almost all of the enzyme kit, analysis kit that involves analysis of starch. So alpha amylase is an endoenzyme. So it goes inside of the um, molecule, the starch molecule, and break not peptides, sorry, bonds, glycosidic bonds. It breaks glycosidic bonds within the starch molecule, inside of it. It's called endoenzyme, or yes, it's an endoenzyme. Exoenzymes, so if we look at the glucoamylase, which is also a very essential enzyme in any of the kits that you get for starch analysis, what it does, it's an exoenzyme. So it takes one glucose unit at a time. It breaks it off the polymer, one glucose at a time from outside. So it's an exoenzyme. Now, polynase and beta amylase are very important enzymes in measuring degree of gelatinization and measuring degree of retrogradation. Alpha amylase and glucoamylase are important enzymes in analyzing total starch, amylose to amylopectin ratio. Beta amylase and polynase are very important for measuring degree of gelatinization and degree of retrogradation. Now, what are they? Polynase is an enzyme that specifically cleave the alpha 1,6 bond, which is the branching spot. So alpha amylase targets alpha 1,4 glycosidic linkage. Polynase targets specifically alpha 1,6, the branching. Alpha amylase can also break alpha 1,6 too, but polynase is specific for the debranching. Beta amylase is also an exo, but it cleaves two glucose units at a time. So it gives you maltose. So it breaks two at a time from outside. Okay? So you need to remember those differences. So beta amylase and glucoamylase are exoenzymes. Alpha amylase and polynase are endoenzyme, but polynase is specific for alpha 1,6 linkage, which is the branching point, and beta amylase is specific for cleaving two glucose units at a time. Okay. One other thing I want you to remember is beta amylase and polynase have zero activity on raw starch, raw or retrograded starch. They cannot hydrolyze. Okay, the starch needs to be gelatinized in order for beta amylase and polynase to actually cleave. So this is important because it's the same. If you remember that question at the beginning of the lecture last time, I said, is the same method used to measure both gelatinization and retrogradation? Yes, it's the same method because it uses beta amylase polynase. Because the more starch is gelatinized, the more these enzymes are going to work. The more starch is retrograded, the less these enzymes are going to work. And we'll see that method later on. Okay, so you gotta remember these differences. Okay, so back to this. Once we break the starch now into glucose, so the entire starch molecule is broken into glucose, mainly you use a kit where you gelatinize your starch, you break it down by alpha amylase and glucoamylase, and then you end up with glucose. Then at that point, when you end up with glucose, you can use an enzyme assay specific to glucose. This is another enzyme assay other than using hexokinase. Here you're using glucose oxidase. In this reaction, you convert the glucose into gluconolactone, and you produce a peroxide. The peroxide is what is important for the second reaction. So this is again a coupled reaction. You have the measuring reaction with glucose oxidase, and then you have the indicator reaction where you use peroxidase. So here your peroxide reacts with the dye or oxidizes a dye in the presence of peroxidase, and you get an oxidized dye that is colored, and you measure the absorption at a specific wavelength. Okay, so, and this all comes in one kit. So you get the starch, total starch kit, that you would have all the enzymes you need to hydrolyze the starch, and then the two enzymes you need for the second reaction, for this reaction to determine amount of glucose, and you back calculate amount of starch. Okay, so I will stop here for today, and I'll finish carbohydrate analysis on Wednesday.